so let's go ahead and get started. A couple of quick announcements. I'm still wrestling with D2L quizzes in order to actually get your scores. All of your answers are there, but the quiz thing doesn't want to talk to grades and vice versa, so we're still working on that. Anyone actually have a score for their quizzes on D2L? Anybody looked? No? Anybody care? Yes, so I am working on those things, and I will try and take care of that ASAP. The other thing that I will try and do is get some audio-only recordings of some of the lectures. I know it's currently on YouTube, but there are a few audios, and I know some people like to have just the audio. So I will try and get those done, again, hopefully today. We only got one more week to go. Eek. What? Nice? Eek? Okay, wait a minute. Yes and no. Probably depends on your point of view. Which reminds me, the final exam is on a Tuesday, the Tuesday after next week from Tuesday at 8 a.m. right here, yes. And don't forget what's happening over the weekend. Time changes, so it's going to feel like 8 a.m. on Monday morning, just to make the last week even more fun. So, but we're going to be talking about techniques. It's going to be awesome. It'll be a blast. Okay, any questions about sort of, last week, last stuff? As usual, week from today will just be review. There will be clicker questions there. And given the problems with D2L, I'm just going to do clicker questions here rather than trying to do online stuff as well. Okay, questions, comments, worries? No, not at this point. Anyone who is trying to use the testing center on Tuesday and haven't signed up there already, it's a problem. It's completely full. So we're trying to set up some other alternative testing solutions for people who need some more time. But 2 hours, so you've got 2 hours, by the way, it's from 8 to 10. So it should be plenty of time to be here. OK, so everyone got your clickers? Speaking of clickers, today, the action of the trip repressor with tryptophan is an example of a positive feedback loop, a negative feedback loop, a flip-flop device, a feed-forward loop, or everybody's favorite, A and B. $30 seconds.
Yeah, I know. I wish I got it earlier.
10, 5. Okay, we're at 92. That's pretty good. So what does tryptophan do? Well, let's back up a little bit. What about the trip operon? Why do you have the trip operon? What does it do?
It produces enzymes to metabolize tryptophan when you're low.
Okay, it produces enzymes to actually make tryptophan when you don't have very much tryptophan around. So when do you want to turn those on? When you don't have tryptophan around. If you have tryptophan around, what do you want to do?
You need it repressed.
Shut it down. So what does that sound like? It sounds like a negative feedback loop to me, yes. So positive feedback loops will be activators that are continuing to activate. And here, this is a negative feedback. It's not their classic negative feedback, as we talked about last time, which is a repressor protein, which is repressing its own synthesis. But it serves in a very similar fashion. Flip-flop device we haven't really talked about, but those are when you've got repressors repressing different repressors relative to each other. So you can change states. You can flip back and forth. And very often you'll have one of these flip-flop devices, which then actually will lead into some kind of positive feedback loop. And we did talk about that in terms of lambda repression, the lambda C1 repressor gene versus the lambda CRO gene, as far as that's concerned. So that's this one. Our next one for today, master genetic regulators such as ILES are most likely to be enhancer binding proteins, silencer binding proteins, coactivators, co-repressors, or chromatin remodeling complexes. $30. 20. 10. 'Kay, what do we think? Most people think that master genetic regulators are coactivators. Unfortunately, I don't agree with you. But why wouldn't I agree with you? Well, because it's wrong. Yes, that's correct. But why not?
Is it that coactivators interact with another enhancer protein rather than directly with the DNA?
So coactivators are going to be interacting with different proteins, and that leads us to are coactivators likely to be general or specific? General. Likely to be much more general than they're likely to be specific. What about these master regulators? We said the nice example of ILES. Really specific. And so those are going to be binding to things down, usually enhancers. It turns out there are a couple of those which are silencers as well, but ILES is an activator, so it's going to be binding to an enhancer. So it is enhancer binding protein. And almost all of these are going to be enhancer binding proteins. Any of these master regulators that are causing, if we think about the now 3, even though I've told you 4 until my last lecture, of those master regulators that are important for getting induced pluripotent stem cells, those are all enhancer binding proteins. Okay, so today we'll see how far we get. But most of this is everything that happens after transcriptional initiation. And how do you regulate the activity of genes after you get that specific start, your pre-initiation complex is assembled, you start to make your first nucleotides. Sort of varies a little bit. Attenuation is actually too early termination in transcription. And we'll talk about how some of that works. Just a particular example, some riboswitches, but that's only one of the mechanisms. We've already talked a little bit about alternative splicing. This is, of course, after you've made your messenger RNA, what pieces end up being in your final messenger RNA. Hadn't talked about alternative tailing yet. It's where you end up with the poly A tail, which, of course, can change the protein that you end up with. RNA editing is a really cool mechanism which was only discovered literally a couple of years ago. Of course, in studying viruses, because virus defense mechanisms. But it turns out RNA editing is also absolutely critical for expression of certain genes. And why you want to edit something after it's been made is still a question we don't quite understand. Probably gets back to it works, and so it's been maintained through evolution. A little bit about alternative export, and the main example here, of course, is viruses, leading on to next term. Messenger RNA localization. When we talked about even skipped, we talked about bicoid and some of the gradients of some of these activators. Turns out that's all due to messenger RNA localization. And so where the messenger RNA is going to determine where you get expression of certain genes. So things like IRES or even skipped or any of these other things, which is really important. Then, of course, once you've made your messenger RNA, how much of it you decide to turn into protein and where you decide to start to turn into protein is going to be very important. So translational regulation, a lot of this has to do with EIF2B. And so everyone remember what EIF2 does? Yes? No? We'll remind you it's initiation, eukaryotic initiation factor 2, bring in the initiator tRNA. And 2B is a different protein, but it regulates EIF2. Stability of messenger RNA turns out to be really important as well. Of course, if you've got mRNA around for longer, you're going to be making more protein. Less long, you're going to be making a lot less protein. It's another way to regulate things. And then regulatory RNAs, which are, again, a whole subject in and of themselves, but at least we can talk about them a little bit here. So basically, this is exactly the same thing in cartoon form, for those of you who prefer the graphical form rather than just all written out. But basically, these are all different possibilities. And we're going to talk about basically all of these through the process. But again, attenuation, you stop transcription. And that's a regulated process. It's not like the pausing that we've talked about before. This is literally everything dissociates. If you mess around with capping and splicing, very often you'll have non-functional sequences, or sometimes these will be sequences which are a little bit different. And usually, as long as this stuff all stays in the nucleus, because translation takes place in the cytoplasm, you're never going to be getting a product of that gene. On the other hand, once you get these things out, you can turn on or turn off. And so this is getting back to what we talked about, right? When we talked about gene regulation at the very beginning of the gene regulation lectures. Transcriptional control is great because it's really early on in the process, and so it's most energetically efficient, but it's slow. It takes time, and that's partly why all of these circuits that we talked about work. Positive feedback, negative feedback, flip-flop, because it takes time between actually regulating transcription and finally getting an actual gene product. So these regulatory mechanisms are a lot faster in terms of turning things on and turning things off. And then, of course, what we're not going to talk about, what we have already, are the covalent changes, the phosphorylations, sometimes the GTP hydrolysis, which is not completely a covalent change, which is a very rapid process. So let's talk about attenuation. Attenuation, again, all this is premature termination of your transcript. And the RNA polymerase has started. It's started to make an RNA, and it's supposed to be here in black. Quick review question. Why did I try and scribble this out here? What's that supposed to represent? So this is the RNA polymerase. It's got the C-terminal domain. But riboswitches are almost always acting in bacteria. Do bacterial polymerases have C-terminal domains? No. So that's why it shouldn't have been there. That's why I chopped it off down here at the bottom. You may see the little square. So all this is, again, it's just the polymerase stopping too soon. There are lots of ways that this can happen, but probably the best understood of these are these so-called riboswitches, which are sequences of RNA which can form very specific three-dimensional structures. And those three-dimensional structures can either lead to transcriptional termination or non-transcriptional termination. So how does termination usually work in bacteria, at least the only kind we've talked about so far? Transcriptional termination. What is it?
Doesn't it cleave itself off?
It's the bump in the oil, right? So you've got a hairpin loop that forms and a stretch of U's. Well, what's happening here? We've got a hairpin loop structure and a whole stretch of U's. So if this structure forms, you'll get transcriptional termination. But it turns out that only forms when this folded RNA interacts with something. And in this particular case, it's guanine, but there are lots of different riboswitches that can bind to lots of different things. So the riboswitch is just an RNA sequence which folds up into a particular structure and then can interact with something. And that stretch of RNA is called an aptamer. That's why I have it up there on the slide. And this is a really fascinating process because it depends on 0 proteins. So people talk about this, again, the book talks about it as being sort of a relic from the RNA world. So this is how RNA can actually recognize things and then change other aspects of what's going on with that RNA. And all it is binding and changing the structure of the RNA. Yeah?
So it's just interacting with something as opposed to necessarily resulting in termination?
Yeah, so the question is, it's interacting with something and not necessarily resulting in termination. So that's exactly true. These aptamers can interact with something. They will change the structure of the RNA that they're still connected to. That could lead to termination, but it could also lead to a number of other things, and we'll talk about those a little bit later on. But it's just binding to a specific ligand, changing the structure of the RNA, and that changing of the structure of the RNA leads to some other effect. Could be attenuation like you have here. Yeah?
The riboswitches are being transcribed by the RNA in ways that they then interfere with? They aren't like independent chunks of RNA that interfere with that?
Yes, so the question here is, are the riboswitches actually made as part of that RNA? And the answer is yes, exactly. So the riboswitch is made as part of the messenger RNA. It's not something that's acting in trans, it's always acting in cis. It's always right there. And that means that you've also very often got, and we'll talk much more about this later, there's also what's called a 5' UTR, or untranslated region, because it's doing something else as opposed to being translated and ending up being made into protein. So that's the first of these things. Attenuation, there are other ways of doing attenuation as well. It's just premature termination. Premature termination stops. You're not going to be making any more. We talked about alternative splicing quite a bit already when we talked about splicing in the first place. Just as a quick review, the more complex, as they put in the textbook, all of my yeast biologist friends say just it's a bigger organism as opposed to a more complex organism. Of course, I like to think that as a microbial biologist as well. But in the larger organisms, usually have a lot more regulated splicing than you do in some of the smaller organisms. And basically what that means is, even though the number of genes, protein coding genes, is not that different between smaller organisms and larger organisms, very often in those larger organisms you have a lot more possibilities in terms of alternative splicing. There are a couple of different mechanisms that you have for alternative splicing, and basically, just like we've already talked about for regulation, probably ad nauseum now, positive and negative. So positive, you stimulate usage of a particular splice site, and negative, you block the usage of a particular splice site. And because of this alternative splicing, it means we really have to kind of redefine what people used to call a gene. The original Beadle and Tatum was one gene, one polypeptide. Well, if you've got multiple different splicing products, and the example down here at the bottom is the DS-CAM gene from Drosophila, which can have over 36,000, if I remember correctly, different possibilities in terms of what can end up being spliced from one stretch of DNA. They're always related. They've always got a lot of sequences which are the same. And so here, you look at the gray segments down at the bottom. These are what's called constitutive exons. You always use those. It's just the colored ones which are different relative to each other. So alternative splicing happens a lot, particularly in larger organisms like us. I wanted to give a particular example here. Unfortunately, this is one which got cut from the 6th version of the textbook, but it's in the 5th version of the textbook. If anyone wants to borrow it, you can take a look at it in a bit more detail. I think it's also in the one which is available online at NCBI Books. But here, this is a nice example of how splicing regulation leads to something actually, at least for fruit flies, is rather important. whether you're a male or a female depends on alternative splicing. And two different kinds of splicing regulators. There's a repressor of splicing and an activator of splicing. So I think this is a nice example of looking at that. These proteins, let's start down here at the bottom. As always, these are geneticists who discovered these in the first place. So if you have a mutation in this gene, you name that gene after the phenotype. And so the best one here down here is the bottom, DSX, which just stands for double sex. So if you don't have this gene, the flies develop as if they have two different sexes, or at least the sexual characteristics then end up being both. They're actually not fertile. But then the genes which regulate double sex transformer, so you have mutations in these, the sex is transformed between the two, and then the master regulator in this case is a splicing regulator. It's called sex lethal. And so if you have mutations in this gene, turns out all the female flies die. But the males do just okay. So interesting aspect, and this was work that was done 2 floors down from where I did my PhD. When we had our seminar speakers, there'd be fruit and a few flies floating around the fruit basket, and it was, oh, that's one of my mutants. Look, that's one of my mutants. Geeks. What can I say? So, but the way this works is sex lethal, as I mentioned before, is a splicing repressor. And so, and it regulates its own gene. So we have here this feedback product right here. So in the presence of sex lethal, it blocks splicing, which would normally happen right here. This is what happens in the male lineage. Blocking splicing, you don't make the sex lethal protein, at least not a functional one. Here, however, if you block this splice site, once you've blocked one of these 3' splice sites, everybody remembers U2, U2AF, interactions here. You've blocked the binding of U2AF, no binding of U2, no splicing there. So then you just go on and find the next one, because this is all happening as these things are transcribing. Now you make this functional protein, and you have a positive feedback loop here, where, yes, it's a repressor, but the presence of the splicing repressor leads to production of a lot of this particular protein. This repressor then will bind to the messenger RNA from the transformer gene, and in an extremely similar process will now block these 3' splice sites, also leading to a non-functional protein in the males, because this splicing is normal. If you block that splice site, you now have a functional protein. This now leads to the protein actually does the job down here at the bottom, which is double sex. And this is a transcriptional repressor. It again has different splice sites, but now these transformer proteins lead to activation of this 3' splice site that's otherwise not used. And what that does is it leads to a different C-terminus on this transcriptional regulator. That different C-terminus is your DNA binding domain. The DNA binding domain is now going to be different. So for your male ones, you have a male-specific DNA binding domain. For females, you've got the female-specific, and these guys block male differentiation genes if you're in a female, female differentiation genes if you're in a male, which if you have a lack of this gene, you don't repress any of those genes, so you end up with the double-sex phenotype. So these are splicing regulators which are leading to this particular phenotypic difference. Yeah, in the back. So, okay, the question is, does this all take place on the X chromosome? One of the things that I'm avoiding here is talking about sex determination in fruit flies, because it's a lot different than sex determination in mammals. So the sex lethal gene, actually I can't remember which chromosome it's on, but these are all on different chromosomes throughout the fruit fly. And so it's not one specific chromosome where this is happening. All of these proteins are made, and once they've made, they can diffuse anywhere. And in fact, they're all being made in the cytoplasm. And so you will make this messenger RNA. That will then get translated out in the cytoplasm. That protein comes back into the nucleus. Where that protein ends up going could be anywhere. It could be any of the different proteins which are, sorry, any different chromosomes which are there and how they're being transcribed. This is true for any protein which is being produced. Once it comes back inside the nucleus, it's going to be acting in trans. Okay, well, there's XX, YY, and that's getting way ahead of us here. If you want, we can talk about it offline. It's a really fascinating process. I think it's really cool, and the X to autosome ratio and how that works. But yeah, be careful about the internet. It's wonderful, but it can suck you down rabbit holes if you aren't careful. Okay, so that's what happens with splicing. It's just one example, probably the best understood of these examples, partly because of Tom Klein and the work that he was doing at Berkeley. And SRY is a really interesting question about that, too. But then if you think about the next step that happens during messenger RNA formation, that's going to be addition of poly A tails. And again, all this is co-transcriptional. As you're making your messenger RNA, you're splicing it, you're chopping it, you're adding a tail. Poly A tailing is dependent on basically two major proteins, at least in terms of specificity. Where do you have them? What are those two proteins? One of them's up here. That's easy. CSTF. What about the other one? CP something F, yes. CPSF, cleavage and polyadenylation specificity factor. Yes, this class is alphabet soup. Those two proteins, and it's the concentration in particular of CSTF, which determines whether you will actually cut a messenger RNA as it's being made and make a poly A tail there, or not cut that particular messenger RNA and make a poly A tail there. And so in a case where you have low amounts of CSTF, you keep going past this particular stop codon, goes along, CSTF will keep going here. You've got splicing that takes place. The stop codon is gone. Now you end up with a poly A tail over here. This gives you a different C-terminus than here. We have lots of CSTF. Remember, all this stuff is happening simultaneously. You've got a lot of CSTF. As soon as you transcribe a potential cutting site, you can get cut, maybe even before it gets spliced. And so that's what happens in this case over here. Now you cut, you make a poly A tail. You end up with two very different C-termini. Last time we talked about the difference in the C-termini, last slide I should say, where you get different binding of different repressors. Here it just depends on where your antibodies are. And so this is a figure from this term's text as opposed to last time. Here, this C-terminal region leads to antibodies bound to the cell or secreted antibodies. And just because of that amount of CTSF, and that has to do with the stimulation state of the B cell. And actually, how many of you are taking immunology right now? Okay, I know at least one or two of you are. So you probably know more about this than I do. But at least in the stimulation of the B cells, in order to get them to be secreting antibodies, you end up increasing the amounts of CSTF. And so you're going to make many more of these antibodies with the secreted tail as opposed to those that are stuck down in the membrane. So now we've talked about differential splicing, different splicing regulators, talked about different tailing. Now we've got our messenger RNA. Normally, right, you're going to export this. It's going to get translated or something else is going to happen to it, except and occasionally you change that messenger RNA. And this is, again, this is a really bizarre process, and nobody thought it was happening until they actually studied it in a little more detail, and it turns out, yeah, we do have RNA editing. Turns out you have RNA editing both in mitochondria, but of course we're more interested in mammals because that's what we happen to be. There's one really pretty major one which happens. It's this change of adenine to inosine, and that change, adenine to inosine, changes the base pairing interactions that you would have with, what otherwise would be an A now becomes an I. Where else have we talked about inosine? In the wobble base, so it's in what? So it's in the anticodon, which is in the tRNA. The codons are all in the mRNA, so the anticodon's in the tRNA. So this is now in the messenger RNA where we make the change. But you're exactly right. It's in the wobble position. Why is it in the wobble position? Because it can bind to a whole bunch of different other nucleotides. But now instead of it being the tRNA that can bind to lots of different codons in your messenger RNA, it's the messenger RNA that can bind to a bunch of different tRNAs. And so basically what that does, it changes the amino acid sequence of that particular codon, how it's going to be recognized. Now, of course, you don't want to do this to all of the adenine residues that you have. This would be a god-awful mess. So you have a very specific adenine deaminase that happens on RNA, so that's what this ADAR enzyme is, that will bind to a specific secondary structure and then modify one adenine that it happens to be in the middle of it. And we know that this ADAR enzyme binds to double-stranded RNA. It's been well characterized, and so again, it has to be a secondary structure, which is forming a messenger RNA. Also tells you that yes, messenger RNAs are not just unbase paired single-stranded pieces. Very often they're double-stranded as well. Yeah.
What does this change do? And can you remind me again when we have it in the when we learned about it originally? Does it create a stop?
So what does this enzyme do, or what does adenine to inosine do?
The second.
Right. So adenine to inosine, in the case of the tRNAs, that's one of the modifications that happens, which is important for getting fewer tRNAs. Because again, normally if you had to only your normal base pairing interactions, then you'd need way more tRNAs than you actually have. And so that's the whole idea of the wobble. And that inosine is in some cases put in by an ADAR enzyme, but there are other modification processes that happen there as well for tRNAs. Because this happens all over the place. Here, this is actually a very specific mammalian change, and these adenine deaminases are very specific to mammals, whereas you have inosine all throughout life in tRNAs. This change to inosine is a very specific one that you just get in mammals. And it turns out this particular change was not surprising. It changed the amino acid sequence, and that change in the amino acid sequence actually is absolutely critical for brain development. So if you have people who have defects in this particular gene, they have, and actually usually don't even survive to term. So you actually have to have this, and again, it's evolutionarily speaking, it's been there. Yeah?
And this is mRNA, it looks like?
This is a messenger RNA, exactly. And has to form a double-stranded structure in order to get this modification taking place here. Oh, yeah?
You said this is very critical for brain development. Is this only being expressed then in neurons that are receiving the signal to differentiate, stem cells that are receiving the signal to differentiate into neurons? Or is it in all cells in the developing?
So the question is, how specific is this to neurons? I think that this particular modification, this is just one example of where you clearly need it. I'm not absolutely certain about that, when exactly you're getting this particular ADAR being expressed. I know a lot more about these adenosine deaminases, which seem to be important for protection against viruses. And so there are a number of those which are being expressed actually pretty generally. For the particular example for brain development, I don't know the exact details. So I also mentioned that you can get U sequences which are added. Sorry, back up here. Mostly in mitochondria, but you have some in cellular genes as well. Apolipoprotein is just a nice example of that. And this isn't a good example of where you do have differences in terms of expression in different cell types. Here we'll have the editing that happens in liver cells Actually, it doesn't happen in liver cells. You have your normal protein, which is made, and then editing that happens in intestinal cells, and you end up with a stop codon, which is being made. And again, U is going to be, you can find it all over the place, but UAA is a pretty common stop codon, which is being used in this particular case. So it seems that somehow, again, evolutionarily speaking, you had a modification to make a stop codon in this particular gene, and that turned out to be a competitive advantage, so those ended up winning out. So 2 examples of RNA editing. Again, one which is critical for recoding, and this one is giving you different stop codons. So, okay, hopefully now we've got our edited or non-edited RNA. Now we need to get it out of the nucleus to get it translated. Turns out that, again, not terribly surprisingly, this is regulated as well. And again, this is best understood in looking at viruses, particularly our friend HIV-1, which I may be getting some into my lab, but probably not until well after this term. I've got to go through all the safety procedures to do that as well. I currently don't work with HIV-1, but I may be. So HIV-1 is a really fascinating virus. We talked about it in terms of retroviruses before. Comes in as a single-stranded RNA, then gets reverse transcribed into double-stranded DNA, which gets inserted into the genome a lot like all of the virus-like retrotransposons that we have in our genome. Again, 8% or so of our genome are these kinds of things. Once it's in the genome, then that DNA gets transcribed. Gets transcribed into multiple different RNAs. So there either be the RNAs which are going to make the viral proteins, but really importantly, you also have the RNA which is going to be put into the virus. And that RNA has to be everything, whereas all of the RNAs that you need to make the proteins have to be spliced. So somehow there has to be a way for that non-spliced RNA to get out of the nucleus, get packaged, and end up turning into a virus, so it can go out and be infectious again. So how this works in HIV is there's a particular protein called Rev, and Rev is made through a spliced messenger RNA, which you can see up here at the top. This is that Rev protein. It gets spliced. The messenger RNA for this gets spliced. It gets exported into the cytoplasm, gets made, translated in the cytoplasm, re-imported back into the nucleus. But once Rev comes back into the nucleus, it binds to a sequence of RNA which is otherwise spliced out in this Rev protein. So it only binds to this RNA which doesn't have splicing that's taken place in it. And that Rev binding to what's called the Rev response element. What are elements? Elements are always sequences, exactly. So binds to the Rev response element, and now that complex will get exported just by itself. And so that basically pulls your full genome out into the cytoplasm where it can be put into virus particles and continue. And it seems that the most regulated RNA export, not just virus RNAs, but other RNAs that have differential splicing, is mostly to the presence of a protein which will come back in and then help move out those non-spliced. Because you remember, there's all kinds of quality control to make sure that splicing is taking place. We've got our exon junction complexes, we've got our SR proteins, we've got our HNRNPs. All of these are normally used to say, hey, this hasn't been spliced yet. It needs to be kept in the nucleus. So again, a couple of examples like that. But, okay, hopefully now we've got the right messenger RNA. It's been transported out of the nucleus. Where do we put it? Well, most of the times you're going to translate it, right? It comes out of the nucleus, wants to be translated. But in a few cases, and again, like the case for the Drosophila embryos, where you want to have messenger RNAs in one particular place, and in the case of the Drosophila embryo for bicoid, you put the messenger RNAs into the egg even before it's been fertilized, and they basically just sit there and wait for fertilization to take place. And upon fertilization, they then get translated. Well, that's great, but if they're spread all throughout the nucleus, you're not going to have this nice gradient, which is what you need to set up the front end versus the back end of your embryo, get all the expression of your master regulators, etc. So how do you get these messenger RNAs to one particular place inside the cell? And it turns out that's almost always happening in the 3' UTR. So UTR stands for what? Untranslated region, exactly. And 3' is going to be down at the end. Which end? The end after the stop codon. So after the stop codon and before you get to a poly A tail. So a lot of people think that when you think about a eukaryotic RNA, messenger RNA, stop codon, immediately put a poly A tail on it. No. There is almost always hundreds to sometimes thousands of nucleotides which are made after the stop codon, which are important for things like this, and as we'll see, translational regulation as well. So there are lots of different ways that this can happen. I'm not going to go through them. Basically, this is the experiment down here on the left-hand side. If you make a messenger RNA that doesn't have a 3' UTR on it, which is encoding a green fluorescent protein, the same protein now with the UTR that's now with a red fluorescent protein, that localizes to exactly where it's supposed to be. And so just by mixing and matching these 3' UTRs, you can direct messenger RNAs to lots of different places in the cell, and that's why people talk about this as being sort of the zip code of your RNA. Depends on what that 3' UTR sequence is about. Okay, we've got our messenger RNA. It's been spliced, it's been tailed, it's been edited, it's been exported, it's in the right place. Now we need to translate it. So this is the next level of control. There are multiple different ways of controlling translational initiation. Turns out you can also control elongation and termination as well, but we're just going to concentrate on initiation. These are all examples from bacterial messenger RNAs, but basically the same thing works for a lot of eukaryotic messenger RNAs as well. And the idea here is basically you just want to block binding of the ribosome. So binding the ribosome in bacterial systems is binding to the Shine-Dalgarno sequence, so it's that small subunit RNA sequence which is complementary to a sequence on your messenger RNA. That's shown here as the orange region on each of these. So here, our orange region is not hidden from the rest of the translational machinery, so it can be translated. Sometimes you'll have a protein which will bind to not the Shine-Dalgarno sequence, but usually a sequence right next to it. Again, secondary structure, hopefully secondary structure, or base pairing in RNA. It's going to be a theme which is going to come up again and again and again and again and again. So that's why I don't like single-stranded RNA. You can have riboswitches down here at the bottom. So it's getting back to your question about riboswitches and whether it's just important for attenuation. Here's a nice example of a riboswitch where, again, it's a RNA sequence which is made in cis, part of your messenger RNA, folds up into a particular structure, and those structures have functions, and that function is to bind to usually a small molecule, changes the structure of the RNA, and the changing of the structure of the RNA right now basically puts the Shine-Dalgarno in a space where it can't be used by the translational machinery. Probably one of my favorite examples of one of these sequences are these what's called RNA thermometers. And literally what these are, they're RNA sequences that don't bind to a specific ligand, but at a particular temperature, they will change their structure. And so the change in structure at a particular temperature will either allow translation to take place or block translation from taking place. And again, guy was a postdoc when I did my PhD at Berkeley, has been studying this a lot and looking at all the different structures here. But it turns out just 1 or 2 degrees Celsius change can change the RNA structure here in order to get translation or lack of translation. And so if you think about heat shock, when you talked about heat shock again with Eric Iverson, differential sigma factors in some cases, but in some cases, these RNA thermometers are what's being used in terms of getting. And then, of course, this would be now some kind of heat shock protein gene that's important for survival under those elevated conditions. And so this is, I think, a really elegant way with a very small amount of genetic material. You don't need a protein. You don't need a riboswitch. You don't need any kind of extra ligand. All you need is a change in temperature to regulate the expression of this particular gene. And then the last example here, and we'll talk much more about this in terms of small RNAs, maybe next week, maybe towards the end of today, are these antisense RNAs. So this is an RNA which is acting in trans. So it's a short RNA which is being made somewhere else, which has a complementary sequence to the sequence of your messenger RNA. And if that complementary sequence happens to overlap with the Shine-Dalgarno sequence, of course, now the ribosome can no longer associate with this. So antisense RNA, just opposite strand is going to base pair there. It's going to block any kind of interactions. And again, not surprisingly, then you're not going to get translation as that takes place. This, again, these are all examples with Shine-Dalgarno sequences that we have here, but AUG sequences that you have in eukaryotic messenger RNAs can be regulated in very similar ways, although I don't know of any that have these thermometers yet. Wouldn't surprise me if there are some, but usually we, being endotherms, we regulate our temperature much better. So unlikely to be using this particular mechanism. All of these other mechanisms, however, are in fact used for translational regulation. These, however, are all, really specific. They're going to depend on specific sequences, specific messenger RNAs. On the other hand, sometimes you want to just shut down translation completely, or at least slow it down a lot. And when do you want to do this? Under conditions of stress, lots of DNA damage, or those nasty V things which are happening. And so here, you want to shut down all translation. And the reason behind this is basically, as we talked about before, and hopefully I talked about enough that you're remembering it, translation is extremely energetically demanding. So you've got to make lots of amino-acylated tRNAs. You've got all of your translation elongation factors that are hydrolyzing GTP like mad. So you end up burning lots of nucleotide triphosphates when you're undergoing translation. So if you're under a stressful condition where you don't want to be using energy, shutting down translation is a really good idea. But you don't have regulatory elements for every single one of the messenger RNAs which are there. It'd be nice to have a much more general mechanism. And so that's what this mechanism is. This is regulating EIF2. And as we mentioned before, EIF2, eukaryotic initiation factor 2, it brings the initiator tRNA together with the small subunit of the ribosome to the cap, where it's going to scan along until it finds the appropriate place to start. So EIF2, just like, again, many of the translational elongation factors, is a GTPase. So once you have the appropriate interaction, GTP hydrolysis takes place. You have GDP-bound EIF2. That then goes off, finds another initiator tRNA, and you do this whole cycle again. The problem is, in order to do that, you have to get rid of the GDP and put in GTP. And I think we talked about this a little bit before, but many cases you have to have another protein that does this. It's called a guanosine nucleotide exchange factor. And that, for EIF2, is AIF2B. So it will bind to EIF2 with GDP just after it's started your whole process. Once that happens, it will exchange GDP for GTP. Now we're good to go. We can do translation. Everything is happy and wonderful. On the other hand, if you phosphorylate EIF2, it can no longer interact with EIF2b. And actually, it just does not interact, but it just blocks the activity of EIF2b. And so now you're stuck. You have your EIF2 connected to GDP. You can't start new translation. And so this is a great way of saving yourself a lot of energy because you're not starting new translation. And it turns out that many of stress conditions, virus infection, and even quiescent cells. So a quiescent cell, which is sitting in the G0 phase, which many of our cells are, don't undergo lots of translation, and that's because they've phosphorylated their EIF2. And so they're basically blocking translation, not spending a lot of energy, not moving on through the cell cycle. Yeah?
Is there any chance of those being degraded as long as they're not used?
So the question is there any possibility of these things being degraded? So with these particular proteins, the elongation factors, the initiation factors, et cetera, they're usually pretty stable. The degradation process is usually happening through polyubiquitination, not through phosphorylation. So yeah, they will decay eventually, but usually takes a pretty long time. Yeah?
Does this process also occur in bacteria?
Does this process occur in bacteria? Well, right now, it's talking about E, right? So EIF2. So this is clearly happening in eukaryotes. The process here doesn't seem to be going on in bacteria. It does have a nucleotide exchange factor because IF2 clearly also has GTP hydrolysis, et cetera. But there's a lot less phosphorylation which is happening in bacteria. So it's not this process where it's being regulated. Okay, another part of translational regulation, so-called leaky scanning. And this is, again, getting back to eukaryotic translational regulation. You don't scan when you're a bacterial ribosome. You just bind to the Shine-Dalgarno sequence, and you start from there. On the other hand, if you're a eukaryotic ribosome, you bind to the cap structure, and you zoom along the messenger RNA until you get to an AUG. Turns out that some AUGs are better than other AUGs, and hopefully some of you may remember the consensus sequence that we talked about before. It's not just the AUG, it's also the nucleotides which are next to the AUG, and so those that are closer to consensus are more likely to be used. Those that are not as close to consensus are less likely to be used. So it turns out that how rapidly the ribosome is translocating along your messenger RNA is going to determine which AUG you happen to use. And the way that happens is the concentration of these initiation factors here. There are also some cases called upstream ORFs. What's ORF stand for? Open reading frame. These are usually pretty short protein coding sequences that are in messenger RNAs, and basically what they do is they hold up the ribosome. They block the ribosome. They keep it on your messenger RNA and have it right there, because you remember what happens with eukaryotic messenger RNAs. They end up being these polysomes where your ribosomes are going around and around and around. These upstream open reading frames will bind to the ribosome, get them translating, and then depending on usually the amount of EIF2, that will pop off or keep going and make the rest of your protein. And so usually the rest of your protein, again, it's a much longer open reading frame. It's going to be something which is actually functional. And again, this is getting back to saving energy. Is the longer your open reading frame is, the more certain you want to be to actually translate it. One way that viruses have figured out how to deal with this, annoying process of binding to cap structures and scanning along your messenger RNA is they've developed, or I should say evolved probably more appropriately, these sequences called IRES sequences or IRES structures. This is what's called an internal ribosome entry site. And basically, what one of these irises does is it mimics the structure that you would normally have in a capped messenger RNA. So normal messenger RNAs, caps, poly A tails, this whole complex that forms between poly A binding proteins, EIF4G and EIF4E. Irises are secondary structures that form in your messenger RNA, which bind to some of these initiation factors. In some cases, irises will bind to EIF4G. Some cases will be EIF4E. But all of these lead to association of the small subunit of the ribosome, which can now scan along this sequence until it gets to an AUG. And after that AUG, get your translation taking place. This turns out to be really useful in cases where the cell is reacting to a virus infection by trying to block any of these interactions. Yeah?
So this is for viruses, but it says EIF, so viruses do this in eukaryotes. Why is it viruses and E?
So these are viruses that are infecting eukaryotic cells. So these are eukaryotic viruses that are infecting here. So, and again, we'll spend a lot more time talking about this next term when we talk about some of the eukaryotic viruses that are there. But basically, this is a cap-independent process of getting the ribosome to a particular place in your RNA. And it turns out there are a couple of reasons that viruses want to do this, but it's also a really useful way if you happen to have an internal open reading frame that's, say, in the middle of your messenger RNA that's kind of a long way away from the cap that you still want to translate at some time. And it turns out that there are cellular genes that also have these secondary structures that form in the RNA, which will lead to the ribosome binding there and giving you your translational start somewhere other than right up at the 5' end of your messenger RNA. In order to get translation, of course, you have to have messenger RNA, and messenger RNA is terribly splendid and worthwhile, except that RNA, which is one of the big problems of the RNA world hypothesis, is not terribly stable. And certainly for bacterial RNAs, they're really unstable. Usually bacterial messenger RNAs are only stable for a couple of minutes at most, and that's one of the ways that bacteria seem to regulate their protein production. There's not very much RNA for any given protein, so you don't have that much protein which is actually being made. On the other hand, eukaryotes, probably partly because they have this really inefficient mechanism of making the RNA in one compartment and translating it in another one, usually these messenger RNAs are a lot more stable, anywhere from half an hour to tens of hours. That being said, there are some messenger RNAs, particularly in eukaryotes, where the stability of the messenger RNA is also really important in terms of how much protein is made in any particular condition. And so what happens here is that eukaryotic messenger RNAs, what do you do to them? You cap them, you tail them, right? Why do you cap and tail them? It's to protect your messenger RNA from the nasty exonucleases, right, that love to chew in at the ends of your DNA. And those are the things which, after you've had splicing, et cetera, everything that's still in the nucleus, those get chopped back down to individual ribonucleotides. So the basic mechanism whereby almost all eukaryotic messenger RNAs that get rapidly degraded happen is because you have some kind of endonuclease that happens. So if you have any kind of cutting in the middle of your messenger RNA, that's going to provide a unprotected 3' end and an unprotected 5' end. The exonucleases are just going to go nuts and chew this up, disappears very, very rapidly. Turns out, though, pretty much all eukaryotic messenger RNAs, again, they've got these poly A tails at the end, depending on how long that poly-A tail is, actually is going to determine how stable your messenger RNA is. So even while you're in the cell, poly-A tail, fine, it's bound to the poly-A binding protein, but there's no particular modification. A poly-A tail still has a 3' OH at the end. So that's going to get chewed in by the exonucleases. They're always there. And as soon as they get past this poly-A tail, then you have this really rapid degradation that's going to take place. And that's basically shown here. Here's our little Pac-Man. Anybody remember Pac-Man? Anybody old enough to know Pac-Man? Our little Pac-Man exonuclease here that chews in at the ends. And as soon as it's chewed in at the ends, usually actually you'll have then these endonucleases that take place here. Also we'll have the decapping process. So this is what happens all the time, and eventually is going to lead to your messenger RNA degradation. If you want to have really rapid degradation, you'll have an endonuclease that's going to chop here and provide those ends which are going to get degraded. Yeah?
Is the reason that it's so much slower chewing out the poly-A tail because of those poly-A binding proteins?
So why it takes a long time to chew up the poly-A tail? Some of that's the poly-A binding proteins, which certainly will protect it a little bit, but it's nowhere near as good as the protection that you get from the cap and the cap binding proteins. But yeah, if you have an absence of poly-A binding proteins, your RNA is degraded much more rapidly than in the absence of those. So the last thing I'm going to talk about today are these regulatory elements in terms of how you can have one protein which actually regulates translation in two different ways. So this is the case of, ferritin and ferritin, the transferrin receptor. This is iron regulation in, I think I remember correctly, it's in bacteria, but you've got similar kinds of things going on in eukaryotes as well. This protein called Aconitase is an RNA binding protein. It binds to a secondary structure. Turns out this secondary structure is present at the 5' end of the ferritin gene and at the 3' end of the transferrin receptor. Ferritin basically pulls iron together, and transferrin brings iron inside the cell. So in the presence of iron, because iron's a really, really important metal. You have to have it, but you don't want to have it in too high levels. So it turns out too much iron leads to production of a lot of reactive oxygen species. So you need to regulate the amount of iron inside your cell very, very closely. So one of the ways that happens is through this aconitase enzyme. Again, ferritin will bind up iron so it can't lead to free radical formation, and transferrin brings in iron. So in the presence of iron, you want to make ferritin, and that happens because the aconitase enzyme won't bind to this secondary structure. In the absence of iron, you want to have production of transferrin because it's going to be bringing iron inside the cell. And so this stabilizes your messenger RNA basically because it's not getting chewed in at the end. And so that process is, yes, the poly-A binding protein helps, but binding of aconitase helps even more because that exonuclease is never going to get to the rest of that messenger RNA in order to be able to degrade it. If, however, you've got a bunch of iron around, you don't want to be bringing more iron inside the cell, so this transferrin receptor comes off of the RNA, and this gets degraded much more rapidly. So we'll talk about small RNA regulation on Monday before we get to talking about techniques. Have a good weekend. Don't forget to set your clocks forward.